Okay, great. So now I am live basically. Yes. So yes, welcome back to this channel, and today on this session we will be discussing this cell cycle in one shot with the help of these mind map questions, mind map series, right? So this series is going to help you when your exams are coming or you want to revise this particular chapter in no time. So without wasting our time, let us start with this particular session. So the very first one I would like to start, that is cell division, is of two types. The cell division consists of the mitosis and the meiosis. This we have discussed. Yes, sir. Now, if I talk about the mitosis and the meiosis, which one was the equational division? So the equational division was what? The equational division was the mitosis. Why? Because in mitosis, number of chromosomes, if suppose the parent cell is having n number of chromosomes, the daughter cell will also have the n number of chromosomes. If the parent cell is having 2n number of chromosomes, then the daughter cell will also have 2n number of chromosomes. That means there is no change in the number of chromosomes. And this is the reason why we call mitosis as an equational division. Now, if you need a detailed explanation why it's not changing, then you have to go and watch the detailed lecture which we did in previous days. Now, this mitosis was divided into few parts, okay? But before that, let us talk about this cycle which is present just right behind me. So this cycle was what? This cycle represents the whole phenomena which is happening in a cell cycle. So before entering to the mitosis, cell undergoes some preparatory phase, and that preparatory phase is known as interphase. And this interphase consists of what? This interphase consists of three phases, which is the G1 phase, and the second one was the S phase, and the third one was the G2 phase, right? So before entering into the actual cell division, the cell undergoes preparatory phase, which is G1, S and G2. Combining G1, S and G2, they both, all of three are known as interphase, right? So what happens in G1 phase now? So in G1 phase, basically, organelle duplication takes place. All the organelles such as mitochondria, such as Golgi apparatus, endoplasmic reticulum, they all divide and cell is undergoing the normal growth. Remember that in this case, there is no reproduction and there is no sign of reproduction. Why? Because in G1 phase, the cell is undergoing normal growth. Now, when the cell enters into the S phase, then only it starts preparing for the division. In the S phase, a very major event occurs, and that event is DNA duplication. Okay, and since DNA is duplicated, to pack that duplicated DNA, we need histone proteins as well. So histone proteins are also produced in the S phase. Next, the cell enters into the G2 phase. So from G1 to S, S to G2. And when the cell enters the G2 phase, what does it do? It produces some proteins for the M phase. And what kind of protein? So the name of the protein here is tubulin protein. So cell is producing a tubulin protein which will be required for the M phase. So this was all about the interphase we discussed. Now, after all these phenomena, the cell is entering into the mitosis or meiosis, depending on whether the cell wants to do mitosis or whether the cell wants to do meiosis. So when we go into the mitosis, as we discussed, it's an equational division, and this division, this phase, this M phase is divided into other sub-phases, which was prophase, metaphase, anaphase and telophase, right? Great. Now, If I talk about the prophase, so we have divided this prophase again into two phases, which is early prophase and the late prophase. If I talk about this early prophase, so early prophase, in what happens here, the condensation, the chromosome starts to condense, like they have just started to condense, initiation of chromosome condensation, and the centromeres begins to move towards the what, towards the pole, right? And this follows the interphase. What does this mean, sir? Follows the interphase. See, if a statement comes follows the interphase, that means this M phase is coming after the interphase. If I'm gonna say you that S phase follows which phase, so you are going to answer that S phase follows the G1 phase. In the similar manner, if I'm going to ask you that metaphase follows which phase, so your answer will be metaphase follows the prophase. So this is what the meaning of this particular line which is follows. Now we have seen that in prophase there are late and the early phases. So in early phases everything is gonna start, and whatever the thing has been started in the early phase, it is going to be completed in the late phase. This is how you can learn. So in the late phase, what happens? Mitotic chromosomes, the chromosome is now condensed, it is kind of visible to us, and that type of chromosome, that condition of chromosome, we are going to call it as a mitotic chromosome. Now here you can also see that there are two chromatids and one chromosome. There are two chromatids and one chromosome. Have a look over this diagram. This diagram explains the condition of two chromatids and one chromosome. Remember, we discussed that until and unless the chromatids are not separated, we are not going to consider it as an individual chromosome. So in this particular diagram, the chromatids are not separated, but here we can see this is one chromatid and this is one chromatid. So in total we have two chromatids, but the number of chromosomes is still one. So this happens in the late prophase. Now organelles disappears, nuclear membrane also disappears, and centrosome starts radiating the Astor rays, and these Astor rays are basically what? Basically the microtubules. Great. Now after the prophase, let us move towards the metaphase. So in metaphase, what happens? In metaphase we have this metaphase is basically what? In this, how you are going to understand, like metaphase has been started. So metaphase starts when you are not able to see the nuclear membrane, when you are not able to see anything. At that point of time, you are going to consider that okay, metaphase has been started. The only and only thing which you will be able to see is the chromosomes, and those chromosomes will be the condensed chromosomes, and now you can study the morphology of the chromosome. So from here there are lots and lots of questions which has been asked in the NEET examination, like in which state we can see the morphology of the chromosome. So the answer will be the metaphase. So in the metaphase, we are able to see the chromosomes very clearly. Next, we have in this metaphase also what happens? The spindle fiber is getting attached to the kinetochore, and when the spindle fiber is attached to the kinetochore, they push the chromosome towards the equator and they align themselves. So we can say that in metaphase, all the chromosomes are aligned in the equatorial plate, also known as the metaphasic plate. So this was all about the prophase and the metaphase. Now let's go with the anaphase. And in anaphase, what happens? Chromosome splits from the centromere. So we had what? We had this type of chromosome. Before the anaphase, we have this type of chromosome. We had two chromatids which was attached to one centromere. But in the anaphase, what is going to happen? They both are going to be separated. How they are going to separate? They are going to separate it like this. These chromatids were arranged where? These chromatids were arranged in the equatorial plate, right? So how they are going to be separated? One chromosome is going to go up and the other chromosome is going to go down, right? Now we can say that now these are what? These are the individual chromosomes. Why, sir? Because they are splitted from the centromere. Now if I talk about how it is moving, so you can see the centromere is at the first, so that means the centromere is leading and the chromatids is the trailing end. So the first one to reach towards the pole will be the centromere and then the chromatids is gonna reach. So this is how the movement of chromosomes occurs during the anaphase. Now after the anaphase, which was the main phase of the mitosis, because here only the distribution of chromosomes is taking place. After this, the cell enters into the telophase, and telophase is nothing but the reverse of prophase. So whatever happened in the prophase, you just have to reverse it and write it in your exam. Like chromosomes lose their identity. We saw that in prophase, the chromosomes were what? The chromosomes were condensed, and now we are able to see the chromosomes properly. But now again they decondense and chromosomes is now called as chromatin fiber. They are just like a ball of wool which can be said as the chromatin fiber. So we can't identify the chromosomes here. So they lose their identity. Nuclear envelope, which was what? Which was deformed, which was dissolved in the in the prophase, they are what? They are again reappearing in the telophase. All the cell organelles which were dissolved, they were gone, like Golgi apparatus, mitochondria, which we are not able to see. All of them are what? All of them are again appearing in the telophase. And this is the point where meiosis gets off. After telophase, like what is going to happen? Karyokinesis is completed, and then cytokinesis will also get completed. Now let us move on and let us understand this mitosis with the help of a quick diagram. In the prophase, this diagram represents, these were the centromere, they starts moving towards the poles, right? And now in the late prophase, what happened? The centromere is at the poles and the chromosomes are now properly visible. In the metaphase, the nuclear membrane gets completely disintegrate, the spindle fiber gets attached to the chromosomes through the kinetochore, and in the late metaphase, all these chromosomes are aligned in the equatorial plate. And see, if I talk about the ploidy of these things, so since the chromosomes are what? Since the chromosomes are attached with each other, they will be considered as what? They will be considered as N. But the DNA content is what? Right now the DNA content is 4C. Why 4C? How 4C? What was 2C? What was 2N? All the things we have discussed in our previous lessons. If I'm going to discuss all these things again, this is going to take lots of time. And this session is only and only meant for revision. So if you're not able to understand while you are watching live or while you are watching it recorded, please go back to those sessions, watch that concept, and then again come back and then do this session. Next we have the after the metaphase. Like in metaphase, what's happening? See, in metaphase, we have what we saw, that all these chromosomes are aligned in the equator. After that comes the anaphase, and during the anaphase, what happens? During the anaphase, see, the chromosomes were like this, yes or no? They were attached. Okay, I will show you again. The chromosomes were attached here, both the chromatids were attached, but in anaphase, what is going to happen? They will be pulled by the spindle fiber, and these spindle fiber is going to separate the chromatids, and this is how all those chromatids is separated, and they are giving rise to the 2 chromosomes. So here now we can say earlier they were what? Earlier they were N, and now since they are separated, so they will be called as 2. And remember, we discussed that when the chromatids are attached with the same centromere, then we will call it as an N. And when they are separated from the centromere and they now have their own centromere, so now we can say that this is what? This is 2N. So this is how in mitosis chromatids get separated during the anaphase. And in telophase we all know that it was a reverse of prophase. That's very good. Now let's move on with the meiosis. And in meiosis also, you don't have to remember anything in the meiosis 2. Meiosis 2 is the same as mitosis. Whatever happens in mitosis, it is same in the meiosis 2. The only thing which we have to focus on this session is the meiosis 1. So in meiosis 1 we have the prophase 1, and this prophase in meiosis, the prophase is a kind of very big as compared to the mitosis. So see, in the meiosis we have meiosis 1 and meiosis 2, and in meiosis 1 the prophase 1 is little bit longer than the prophase of the mitosis. So here we have liptoting, zygotine, fecitine, diploating and dikinesis. These are the steps, these are the phases which is present in the prophase 1. So in prophase 1, liptoting is the part where you can see a very thin chromosome. Very thin, very short, long, very thin and very long chromosomes, and this stage is known as Boncased stage. And this is a short-lived stage. That means liptoting is not a very big phase, it just comes and goes. Then here we have the zygotine. So zygotine is the stage where synapses were formed, where synaptonemal complex is formed, where bivalent is formed and the tetrad is formed. So what were these things? Just if you need a detailed discussion about this, we have discussed this in the previous class. Again, I am saying because this session is meant for the revision, so I am not going to discuss all those things in very detail, but I am going to give you a very brief idea here. So we saw that during the meiosis, what happens there is a pairing of homologous chromosome, and the pairing of homologous chromosome is only known as synapse. So then why do we call it as tetrad or bivalency? The term tetrad is basically what? The term tetrad is used for the number of chromatids. So how many chromatids do we have here? So if you see, we have 1, 2, 3 and 4 chromatids. 1, 2, 3 and 4 chromatids. So how many chromatids do we have? We have 4 chromatids. We have 4 chromatids, and this is the reason why we are saying it as tetrad. Next, what do we have? We have the term bivalent. So sir, why do we call it as bivalent? We call it as bivalent because of the number of chromosomes. So how many number of chromosomes do we have? So here in total we have 2 chromosomes. One is this and one is this. So this is the reason why we are calling it as a bivalent chromosome. Clear? Now let's move on with the another stage, which is known as fecitine. In fecitine, these 4 chromatids are clearly visible. When you are going to see it under the microscope, you can see all these 4 chromatids very clearly. Next, what? Next we have there is a formation of recombination nodule and crossing over takes place. Crossing over is a hot topic in this chapter because from this topic lots and lots of question has came, and almost every year NEET has this questions. So let us understand this in a little detail. So here what is happening in the crossing over? In the crossing over we saw that there were 2 homologous chromosomes, like that means one this and this, they were paired, and during the fecitine they just form a recombination nodule and their DNA got exchanged. Some of the genetic material they got exchanged, and this is what crossing over is basically: exchange of DNA material between the non-sister chromatids of the homologous chromosome. This is the crossing over. And what is the need of crossing over? Crossing over is that phenomena from which the variations is happening. So in sexual reproduction, this plays an important role to create variation. And this is also a reason why sexual organisms prefer meiosis in making the gametes. One is the reason that they help to conserve the chromosome number, and the second reason is that in meiosis variations can take place because there is a mix and match of the genetic material from the father's chromosomes and from the mother's chromosomes. Now let us move on and let's talk about the diploating. So in diploating, whatever the things were happened like synaptonemal complex and everything, they get dissolved and there is a new structure which is formed is chiasmata. So in diploating, there is a new structure which is forming, which is chiasmata. Chiasmata is what? It's just a knot-like structure. And this chiasmata also gets dissolved in the dikinesis. So in dikinesis, terminalization of chiasmata takes place, nucleolus disappears, transition to metaphase happens, and nuclear envelope also disappears. So basic thing is similar to the prophase of mitosis, like the condensation of chromosome is happening and the cell organelles are getting disappeared. But two major things in which meiosis is different from the mitosis is the crossing over, and the second one is what? It is the formation of the chiasmata. Next, after this, what happened? The metaphase 1. The cell enters to the metaphase 1, and in this metaphase there is the formation of bivalent chromosomes, and the bivalent chromosomes which is formed in the prophase 1, they all gets arranged into the metaphasic plate. So how, sir? See, this was the one chromosome of the homologous pair, and this was the another chromosome of the homologous pair. So in total, how many chromosomes are? 2 chromosomes, and that is the reason why we are calling it as bivalent, and all of them is arranged in the metaphasic plate in metaphase 1. After comes the anaphase 1, which is a very crucial part to understand. So in anaphase 1, what happens? The homologous pair is getting separated. Don't get confused. Homologous pair is getting separated. The chromosomes, the chromatids are not separated. See, they are going to get separated. They are going to get separated. But still, each of the chromosome is having 4 chromatids here, right? So in this case, what's happening? In this case, only and only the homologous chromosomes is separated. Chromatids are not separated still. Chromatids are going to be separated in the anaphase 2. Now, what happened here is that, see, this both were what? This both were 2n. We were calling them as 2n. Yes or no? Yes, sir. We were calling them as 2n. Now, since they have separated, so this is what? This is now n and this is now n. So this is how meiosis decreases the number of chromosomes. So in anaphase 1 only, this thing is happening. The homologous pair of chromosome is separated, which was earlier 2n, and now they are separated. So they are what? They are n and n, and this will form one cell and this will form one cell. So each of the cell is going to become haploid, which doesn't happen in the mitosis. Now, if I go with the telophase 1, so telophase, again, it's the opposite to the prophase 1. It's not gonna happen that bivalent and all those stuffs is not gonna happen, but just the nuclear membrane will reappear, cell organelles will reappear, everything is gonna reappear. Next, after this, meiosis 2 happens, which is same as mitosis. So let's have a look of the meiosis 2. In meiosis 2, we'll talk about the anaphase 2. So in anaphase 1, what happened? These chromosomes were there, okay, and this chromosome entered into a cell, and that cell undergone meiosis 2. So in anaphase 2, what's going to happen? These chromatids are now going to be separated in anaphase 2. So see, in mitosis the same thing was happening. In mitosis the same thing was happening. See, this was what? This was the mitosis anaphase 1. So similarly we have the chromosomes in meiosis 2 of anaphase 2, and they were getting separated like this. So here also they are going to be separated like this. So there is no point of reading meiosis 2 again and again while you are revising. Leave meiosis 2, concentrate on mitosis 1. Just remember that in anaphase 2 the separation of the sister chromatids takes place. This is what you have to learn in meiosis 2. Rest everything is pretty much similar to the meiosis 1. Now, after all these things, what happens? The cytokinesis. So, cytokinesis takes place in animal cells and plant cells both. So, in animal cell, the cell membrane is flexible, so it can form a furrow and they can divide cell. Yes, that's very good. So, in animal cell, what happens? The cell division is of centripetal like. That means from the outer towards the inward. But have you ever wondered like how the cell division is happening in the plant cell? So the cell division in the plant cell is happening centrifugally. That means something is deposited inside and it is grown towards the outer membrane. So this is known as what? This is known as phragmoblast. What happens here? Phragmoblast is formed. Now what is phragmoblast? They are formed by the help of the Golgi apparatus and some of the proteins, and they grow and they reach towards the cell membrane and they separate the cell into two daughter cells. So this is how the cell division takes place in the plant cell. So here we have revised this particular chapter in just, I think so, 30 minutes. If you need the PDF of this particular mind map, let me know in the comment section, or else you can take the screenshot of this particular thing. I hope that you enjoyed your session. Tomorrow, which is 3rd of October, we are going to start with plant physiology. Same time, same channel. Till then, bye-bye everyone. See you in the next class.